Hi guys, I'm Olivia and we are in the microfluidic lab in Willis Annex, and I'm here today with Bac. Bac, do you want to tell us about yourself and what are you doing?
Hi Olivia, hi everybody. My name is Bac. I graduated from Korea, um, and I work in a Japanese company before joining Tracy Group. And my topic is about artificial kidney. Yeah.
Yeah, so why do you want to look into artificial kidney?
Because most of the kidney failure patients now they relies on hemodialysis.
Yeah.
And hemodialysis has some disadvantage. So hemodialysis is a method require you to come to hospital every 3 or 4 times a week.
Yep.
And treatment gonna long for like 3 to 4 hours at times. And that treatment is not continuous, right? The kidney filtration is continuously within like, let's say, 1 minute, 1 liter of the blood being filtered. So all the toxic substance is being removed.
Yep.
So with hemodialysis, the toxic substance still remains. That's why the people health getting like decline. So hemodialysis keep people alive for like 5 to 10 years. So we want to improve that. And the other thing is because we stick with the patient needs to stick with the hemodialysis schedule from the hospital. So it's gonna be like lower their quality of life because the schedule time.
Yeah, so they have to meet different schedules.
Yeah.
Yeah. So what method are you using for artificial kidney?
Yeah.
What about it that you're doing?
Yeah, we propose new method to do the filtration.
Yeah.
In hemodialysis, instead of using hollow fiber membrane and let the blood contact directly with the membrane.
Yep.
So all the blood cells can be contact with the membrane as well in the filtration process. And in the filtration process, the pressure needs to be applied. So that's why blood cell can be damaged
Yep.
when they go through the filtration all the time like that. So blood cell damage, the membrane can get fouling because of the protein adhesion
Yep.
on the membrane surface.
Yep.
And the other thing can be membrane can make the cell being damaged.
Yep.
And also there's factor of coagulation. When the blood cell contact with the membrane, they're gonna be that's gonna make the defense system works.
Yep.
And coagulation happen. That's why the filtration efficiency decrease.
Yep.
So yeah, we want to separate between blood cells and plasma, and just doing filtration on plasma.
Yep.
Yeah, so we can somehow
Yeah.
get a better filtration. as
So
well.
you're looking at both filtration and separation
Yes.
of the function of the kidney. So essentially, are you using any, what's the setup? Like, do you run an experiment for it? Or-
Yeah,
Yeah.
yeah.
Okay. Do you want to tell us about your
Very
experiment?
interesting question. So what we do is we build a system. You
Yep.
can see in here with the pump,
Yep.
and we pump the fluid from here to the device.
Yep.
And we do the 3D printing for the device, and device with the size of kidney need to function the filtration with the membrane in the middle of it. And we you can see we measure here the pressure, inlet and outlet, and yeah, I can show you the device. So one of our 3D printed device here.
Yep.
Yeah, we have here the O-ring, and we have the micro channel with the spacer we put in, and this one we can improve the filtration efficiency. That's what we studying about. Yeah, and the whole method is how to improve the efficiency of the filtration and how to reduce the damage on the filtration, and how to make the system portable or implantable.
Okay.
Yeah.
So the solution here, because we know that hemodialysis is the filtration of blood.
Yeah.
What what solution are you using here?
Yeah, well, we making some blood mimicking solution for the first stage.
Yep.
So we use glycerol and water and, and satin gum to mimic the blood behavior.
Okay.
And with plasma mimicking solution, we used nanoparticles and some protein
Yep.
to mimic the plasma filtration.
Okay.
So the filtration result here will you see some permeate here.
Yep.
So we will take the permeate into the device and into the container and bring it to the nano side to check the nano particle concentration
Yep.
and also checking with the microscope to see how the fouling happened and how
Okay.
the filtration efficiency increased.
Yep. Hey, Bac, do you want to tell us what this piece of equipment is?
Yeah, so I told you before, we make some 3D printed part for artificial kidney. So we check the microstructure in here of the part. And you can see we put some microstructure in here. Yeah, so with that, we can check through the microscope and we also check the membrane structure as well. So you can see here the structure of the part that I'm presenting. So yeah, so we see the error and we fix the error for the better chip that we want for filtration.
So what's this equipment here? What do you want to tell us something about this?
Yeah, so we do plasma filtration. So we take some sample of what being filtered, and we have some permeate concentration of the nanoparticle inside the solution. So we check the concentration and particle size for the filtration. So we use that nanosite machine. So what you can see is a sample being taken from here and putting into the this chamber, and you can see the laser go down there, and we have the particles in as shown the screen. And when you run the script, run the machine, it can show you the concentration. It can calculate the particle size, and you have the information about how good or bad the filtration is.
So you're looking at the particle size and the concentration from this machine,
Yes.
from the filtration filtration stuff that you have.
Yeah.
Okay,
Exactly.
cool.
Yeah. I
Yeah.
can show you here the running of this. So you see it's recording now.
Yep.
So it asks about temperature and some stuff like that. So after it's recording the pictures, it has count the concentration, and we can see it calculate the particle size here. You
Okay.
can see the particle size about 100 nanometer
Yep.
and the concentration.
So it sounds very comprehensive. Is there any challenges that you're facing?
Yes,
or- yeah.
definitely.
Yeah.
Challenge remain in terms of making the system being biocompatible.
Yep.
So all the material used later, for example, if we put something like this, the chip on the portable device, and we bring it, so all the materials there should be compatible with our blood and our defense system.
Okay.
So it's not making any bad change in the people health. And also the system should be miniaturized, making it smaller and still keeping the function of kidney, which is very hard. The kidney can filter like 1 liter of blood within a minute.
Yeah.
So within 5 minutes, the whole blood in the body being filtered, and all the toxic substances like urea and creatinine being removed. So we want to do the same, but it's gonna be a challenge for us.
Yep. Okay. So how do you think that the research will be further developed? Because it sounds like a very interesting one. So how do you think it can be further developed from here?
Yeah. So if we can somehow improve the challenges, solve the challenges like miniaturization and making the system being tested in animals and human. They're going to be a very good
Okay.
next step for the development.
So putting it back to human being and testing
Yeah.
it for.
Yes.
Okay.